Good morning. My name is Dr. K. Lavanya. I'm working as an assistant professor in Department of Biochemistry, Mallareddy Institute of Medical Sciences. Today we are going to discuss about the biochemistry of cancer. So cancer, as per the definition, it is an uncontrolled cellular proliferation without any apparent relation to the physiological demands of the organ involved. So the cancer can be can be benign tumor and malignant tumor. Benign tumor, they grow slowly by expansion and remain encapsulated in the connective tissue. They do not spread. So it is a benign tumor. In case of malignant tumor, it's characterized by uncontrolled proliferation of the cells um, with cells to various so uncontrolled proliferation of the cells associated with it metastasis. So the incidence of cancer. So etiology, cancers are multifactorial in origin. They are physical, chemical, oncogenic viruses, hormonal and metabolic, genetic and environmental factors play a role. And most common human cancers are spontaneous. So mechanism of carcinogenesis. Um, it is a process where the normal cells are transformed into cancer cells. So the balance between the proliferation and programmed cell death will be lost. So all the cancers originate from one aberrant cell, which undergoes which goes on multiply and produce a tumor mass. So multiple mutations, errors in the DNA replication, failure of DNA repair, abnormal chromosome numbers, all these results in cancer. So carcinogens are mutagens. Examples X-ray, gamma ray, ultraviolet ray, all these damage the DNA by free radical production. So carcinogens can covalently bind to purines, pyrimidines, and phosphodiester bonds of the DNA and cause unrepairable damage. The chemical carcinogens frequently cause mutations by changing the nucleotide sequence of the DNA. So promoters of cancer. Benzopyrene and croton oil on separate application do not lead to development of any skin cancer. But when benzopyrene application is followed by croton oil, then the tumor will develop. That means the croton oil here promotes the development of a tumor. The developmental progressions, developmental of familial adenomatous polyposis occurs by mutation in the APC gene, which can be inherited from the parents and leads to mutations in the KRAS gene, which will progress to form adenomas. Further mutation in P53 gene results in development of colon cancer. Chemical carcinogens are organic and inorganic. In organic, it's a dimethyl benzanthracene, pyrene, and dimethyl nitrosamine are involved. Inorganic, arsenic and cadmium is involved. Occupation, asbestos. Those who are those who are occupations related to asbestos and benzene, they are highly exposed to these substances, so the high chance of getting cancer from asbestos and benzene. Diet. Aflatoxin B produced by the fungus and the drugs, certain therapeutic drugs called carcinogen. It is diethylstilbestrol. Lifestyle changes like alcohol, cigarette smoking, all this leads to development of cancer. Glutathione transferase is involved in the detoxification of various carcinogens, including the cigarette smoking. About 5% of population are lacking in GST. Smokers who are devoid of GST are more prone to develop lung cancers. So here, um, so the chromosomal abnormality and the DNA damage, all this leads to oncogene activation and deletion of oncosuppressor gene. All this leads to malignant transformation of the cells. So anti-mutagens, vitamin A and carotenoids will interfere with tumor promotion and are shown to reverse the precancerous conditions. Vitamin E acts as an antioxidant, prevents the damage made by free radicals and peroxides. Vitamin C regularly given to a person working with aniline prevents the production of new cancer cases. So tubers, beans, and leafy vegetables are shown to interrupt the tumor promotion. Curcumin, the yellow substance that is present in the turmeric, is known to prevent the mutations. So biochemical changes in a cancer cells. The tumor cells display increased metabolic autonomy in comparison to non-transformed cells, taking up the nutrients and metabolizing them in pathways that support growth and proliferation. Tumors have enhanced glycolysis and a suppressed oxidative phosphorylation. It's called the Warburg effect. The tumor cells achieve not only high glycolytic rates, but high rate of nucleotide and fatty acids protein synthesis. So mutated or overexpressed oncogenes and mutated inactivated tumor suppressors influence these activities. Tumor kinetics. So the characteristics of the growing tumor cells. Tumor kinetics in a normal tissue, only 1% cells are in the dividing state, but in the cancer tissues, about 2 to 5% of cells are in a cell cycle. Cytotoxic drugs and radiation will kill the cells in the cell cycle while sparing the resting cells. Here it is a cell cycle. The GO phase is a resting phase of the cell cycle, and from the GO phase, the cells enter into the growth one cycle. From G1 to S cycle, it's a growth cycle, it's a DNA synthesis cycle. Here the P53 and RB modulate the these are the genes which they check the cells. They check the cells. If the cells are good, like DNA is good, then it undergoes. If the cells are good, they go to the DNA synthesis. If there is any like a damage to the cells, these genes stop the multiplication of such cells. Then from the DNA synthesis, it goes to the growth 2 cycle, then to mitosis and cytokinesis. Doubling time, the tumor doubling time in human cancers varies between 10 days to 450 days, but mean is 100 days. That the tumor was present in the body so this indicates the tumor is present in the body for a considerable period of time before its detection. So anchorage dependence. Vinculin is a protein involved in the addition between the cells as well as the basement in the normal cells. Oncogenes stimulate tyrosine kinase, causing the abnormal phosphorylation of vinculin, which results in diminished adhesion as well as the rounded appearance of the transformed cells. Here, the normal cells are of this shape, like oval and round. The cancerous cells, the normal shape of the cells are lost. When your cells continue to grow and divide, the normal shape is lost and there will be variation in shape and size of the cells. Nuclear is larger and darker in the cancerous cell compared to normal cell. And there are abnormal number of chromosomes arranged in a disorganized fraction. So contact inhibition. In a cancer cells, the tight junctions are rare. The property of contact inhibition is lost and adjacent cells continue to multiply to form a multilayer appearance. Cell fusion. Cell fusion plays an important role in fertilization. Fertilization, immune response, tissue repair, and regeneration. It also contributes to the initiation and different stages of tumor progression, including aneuploidy and tumor initiation, origin of cancer stem cells, multi-drug resistance, and metastatic abilities. So metabolic alteration in the tumor cells. Generally, the cancer cells thrive on minimal enzymes. Many tumors prefer anaerobic glycolysis and eliminate the citric acid cycle enzymes. Example, deletion of asparagine synthase in certain lymphomas. So metastasis and secondaries, the collagenase and stromatolin released by most of the cancer cells help in penetration of the cancer cells into surrounding areas. Metastasis is the most common cause of morbidity and mortality. The two types of regulatory genes, oncogenes, anti-oncogenes, and the category of genes that control the cell death or apoptosis are also involved in development of carcinogenesis. So this is a normal cell. Normal genes regulate the cell growth, but proto-oncogenes, these genes that could regulate unregulated cell, so growth and reproduction, and if they damage, like genes that will lead to unregulated cell growth and reproduction, if damaged, oncogenes, these are the damaged proto-oncogenes, which will accelerate the cell growth and cell division. So activation of the proto-oncogenes to oncogenes, viral insertion into chromosomes. When the retroviruses infect the cells, a complementary DNA is made from their RNA by the enzyme reverse transcriptase. The complementary DNA so produced gets inserted into the host genome. The integrated double-stranded complementary DNA is referred as provirus. So activation of a proto-oncogene make to oncogene viral infection results in cancer. Chromosomal translocation. Tumors exhibit chromosomal abnormalities by rearrangement of genetic material by chromosomal translocation. Chromosomal translocation usually results in overexpression of the proto-oncogenes. Burkitt lymphoma, a cancer of the human B lymphocytes, are an example of chromosomal translocation. So gene amplification, administration of anti-cancer drugs like methotrexate and inhibition of the enzyme dihydrofolate reductase is associated with the gene amplification. The drug becomes inactive due to gene amplification, resulting in a several-fold increase in the activity of dihydrofolate reductase. So point mutation, the RAS proto-oncogene is the best example of activation of point mutation, the change in the single base in the DNA. The presence of RAS mutation is detected in several human tumors, that is 90% of the pancreatic tumors, 50% of colon tumors, and 30% of the lung tumors. So, mechanism of action of oncogenes. The DNA damage occurs when this gene becomes abnormal, like P53 gene. It is a tumor suppressor gene. So, when HPV E6 and human papillomavirus E6 and Leigh-Ferrari syndrome, all this inhibit this gene, it's a tumor suppressor gene. When this inhibits, the oncogenes become activated. BCL2 is involved in the apoptosis of intrinsic pathway, and P21 here it's a cell cycle, it arrests the cell cycle. So all this affects the gene transcription and leads to production of a new DNA synthesis. Growth factors. They stimulate the proliferation of normal cells and regulate cell division by transmembrane signal transduction. They bind to the protein receptor and activate the cytoplasmic kinases, leading to phosphorylation of the intracellular target proteins. Here, the growth factor receptors, when there's an overexpression or structural alteration in growth factor receptors, are associated with carcinogenesis. Overexpression of gene ERB, the growth factor, also involved in development of cancer. And non-receptor tyrosine kinases. These proteins are found in the interior of the plasma membrane. They phosphorylate the cellular target proteins in response to the external growth stimuli. The mutation in the proto-oncogene encoding non-receptor tyrosine kinase also increase the kinase activity and phosphorylation of the target proteins, causing unlimited cell multiplication. So these are the oncogenes. These are the oncogenes HER2 are which acts on the receptor tyrosine kinase. This leads to development of breast carcinoma and gastric carcinoma. RET oncogene is receptor tyrosine kinase. This is involved in development of medullary papillary carcinoma for thyroid and phagocytoma. BRCA-ABL is a non-receptor tyrosine kinase. It's involved in development of chronic myeloid leukemia and acute lymphoid leukemia. JAK2, non-receptor tyrosine kinase, it's involved in development of myeloproliferative neoplasms. BRAF here is a serine or threonine kinase. It's involved in development of melanoma, non-Hodgkin's lymphoma, colorectal carcinoma, papillary thyroid carcinoma, and hairy cell leukemia. So these are the growth factors. These are the sources from where the growth factors are produced, and these are the major functions. Oncogenic viruses, mainly here the viruses causing cancer are Epstein-Barr viruses associated with Burkitt's lymphoma and esophageal carcinoma, and human papilloma virus, which is involved in the development of uterine and cervical carcinomas. Hepatitis B virus is involved in development of hepatoma. So Burkitt's lymphoma progression occurs in 3 different steps. The first step, The Epstein-Barr virus, which specifically infects the B lymphocytes, are now immortalized. That is, they can be cultured indefinitely. And the second step of the chromosome translocation. The chromosome translocates from chromosome 8 to chromosome 14. They can take chromosome 14 contains a gene for immunoglobulin heavy chain. Transposing region in a chromosome 8 contains the oncogene c-Myc. And third step is the activation of c-Myc oncogene with consequent malignancy. The Epstein-Barr virus is mainly involved in the development of nasopharyngeal carcinoma. Anti-oncogenes, cancer suppressor genes. The loss of these suppressor genes results in development of several tumors like retinoblastoma, breast cancer, carcinoma of the lung, Wilms kidney tumor. These are the tumor suppressor genes. Retinoblastoma, RB gene present on chromosome 13, Wilms tumor, WT gene present on chromosome 11. Familial adenomatous polyposis FAP, the gene is present on chromosome 5. Deleted in colon cancer DCC, gene is located in chromosome 18. Gene for protein 53, P53 present on chromosome 17. Familial breast cancer, BRAC present on chromosome C, and one representation of chromosome. Genes that regulate the apoptosis are programmed cell death is apoptosis. The gene, namely BCL2, causes BCL lymphoma by preventing the programmed cell death, and overexpression of BCL2 allows mutation of a proteoglycan that can lead to cancer. Thank you.